Take a moment to think about the basic requirements for human survival. At a minimum, we need adequate supplies of food, water, shelter, and oxygen. Microbes aren't that different. In the last video, I discussed the nutritional requirements of microorganisms. In this video, we are going to focus on the environmental conditions necessary for microbial growth and survival. After watching this video, you should be able to discuss the physical conditions that microbes need to survive and thrive. I will be talking about water, temperature requirements, and oxygen needs. Pay careful attention to the vocabulary words in this video. It is a good idea to make yourself some flashcards so that you will be able to remember some of the unfamiliar terms like tonicity, plasmolysis, and capnophile that you will encounter as we're going through this material. We are going to start by talking about the role of water in living things. Water is necessary to support all life as we know it. To review, it is a unique molecule with 2 hydrogen atoms and 1 oxygen atom. The bonds between the oxygen atom and the hydrogen atoms are known as polar covalent bonds, meaning that there is an unequal sharing of electrons between the atoms. These polar substances have partial charges. In the case of water, the hydrogen atoms are partially positive, and the oxygen atoms are partially negative. Because of this, a special type of weak bond called a hydrogen bond can form between water molecules. It is these chemical properties of water that make it so important in biology. It moderates temperature change, it provides a source of electrons for photosynthesis, and it is a great solvent. It is the polar nature of water that makes it such a good solvent. Water molecules can easily disperse other substances that are polar or charged. Water is called the universal solvent because it is able to disperse many more substances than other solvents. All living systems exist within an aqueous or watery environment, where water is the solvent. The cytoplasm within cells contains water, and cells are surrounded by water in the extracellular environment. The term tonicity describes the relative amount of solute dissolved within 2 separate aqueous solutions. We focus on the tonicity of the solutions on either side of the plasma membrane. This is an important concept because it affects the movement of water into or out of the cell. So here I have a picture of a cell. You can see the extracellular environment outside the cell and the cytoplasm. Both are aqueous solutions. They contain water molecules. In this picture, the water's in blue and the solute is the dark dots. In this particular image, the 2 solutions have roughly the same amount of solute. So the term that we use to describe the relationship between these 2 solutions is isotonic. Right? When you have 2 solutions that have the same or equivalent amounts of solute dissolved within the solution, they're called isotonic to one another. It's real important to remember that tonicity is a term that describes the relationship between 2 solutions. And in this class, we're going to be focusing on the relationship between the cytoplasm and the extracellular environment. Okay, so remember that water moves across cell membranes through osmosis. And osmosis is a form of diffusion. Water moves from an area of high concentration to an area of low concentration. In this picture, because we have 2 solutions that are isotonic and the relative amount of water and solute is equivalent to each other, there is no net movement of water across the membrane because there is no concentration gradient. Much of the environment inside the human body is isotonic relative to the cytoplasm within cells. Many organisms live in an environment where the concentration of water is higher outside the cell than in the cytoplasm. This is known as a hypotonic environment. Here, the solute concentration is lower outside the cell in that extracellular environment. Freshwater lakes and streams are examples of hypotonic environments. The amount of solute in these environments is lower than what we see in the cytoplasm. In a hypotonic environment, water moves into the cell through osmosis. As this occurs, the cytoplasm will expand. The pressure on the plasma membrane as a result is called hydrostatic pressure. Bacteria can withstand this pressure because they have an exterior cell wall that prevents that plasma membrane from bursting. Damage to the cell wall, however, can result in lysis or breaking of the plasma membrane. Some protists that do not have a cell wall have a different way of dealing with hydrostatic pressure. They may use a specialized organelle called a contractile vacuole to collect and remove water from the cell. And we talked about those specialized structures back when we were talking about eukaryotic cell structures. The opposite of a hypotonic environment is one in which the solute concentration is higher outside the cell than in the cytoplasm. This is known as a hypertonic environment. Again, remember these are all terms that describe relationships between the two solutions, the cytoplasm inside the cell and the extracellular environment. So here in a hypertonic environment, that solute concentration is extremely high outside the cell compared to what we see in the cytoplasm inside the cell. In a hypertonic environment, water is drawn out of the cell. Right? It moves down its concentration gradient, and this results in shrinkage of the cytoplasm. And the plasma membrane can actually start to pull away from the cell wall. This is called plasmolysis. Here, you can see an electron micrograph of E. coli cells that have undergone plasmolysis. Right? On the left-hand side, you see the normal cells, and on the right, you see the cells where the cytoplasm has started to shrink away from that cell wall. In this situation, cellular metabolism will stop, and the cells can't continue to grow. This is the basis of our ancient food preservation techniques, right? Of adding salt to our food. That, in turn, by adding that salt, we are creating a hypertonic environment, and that can induce plasmolysis in bacteria and can actually prevent microbial growth and preserve our food. Some organisms are uniquely adapted to living in high salt environments. These organisms are known as halophiles. The term halophile comes from the Greek word for salt-loving. Often these organisms actively increase the amount of solute in the cytoplasm to compensate for the high salt extracellular environment. An obligate halophile must live in a high salt environment in order to survive. These organisms are often found in environments like the one pictured here, which is the Dead Sea, or the ocean. On the other hand, an organism that is capable of living in a high salt environment that is but does not require it is called a facultative halophile. Human skin is naturally salty, and many of the pathogens that can cause skin infections are facultative halophiles. So this is a picture of impetigo, and it is commonly caused by Staphylococcus aureus, which is a facultative halophile. Facultative just means it can, but it doesn't have to. And remember, obligate means it's required. It has to have that high salt environment. Staphylococcus aureus, or Staph aureus, is an extremely important human pathogen. One way that we can isolate and identify this organism in the laboratory is to use the selective and differential media called mannitol salt agar. This medium contains enough sodium chloride to inhibit the growth of most bacteria, but it will allow the growth of our halophiles. In this picture, you can see that two organisms are able to grow, but E. coli, which is down here, is not able to grow. It is not a halophile. The color difference that you see in this picture is what allows us to differentiate between Staph aureus and other halophiles. And that color difference is based on the ability of the organism to utilize the sugar mannitol. That's why this is called mannitol salt agar. Another important characteristic of our aqueous environments is pH. Remember, pH is a measure of the hydrogen ion concentration in a solution. You should be able to recall that the more hydrogen ions, the lower the pH, and the more acidic a solution is. The pH of a solution affects the shape and behavior of most of our biological molecules, especially our proteins. For most cells, the pH of the cytoplasm is between 7 and 7.4, which is a neutral pH. Microorganisms can be characterized based on their ability to grow in different pH environments. The optimal pH is the pH that allows for the highest rate of growth. An organism that can grow in a low pH environment, so you can see the blue line here is from this organism can grow from a pH of about 1 to a pH of 5.5 and an optimal pH of about 3. That organism is called an acidophile. On the opposite end of this figure in red, you can see there's an organism that can grow between a pH of 7.5 all the way up to a pH of 11.5 with a peak or optimal pH of about 9.5. That organism is called an alkalophile. Most human pathogens grow best at a relatively neutral pH and are called neutrophiles. So that's the line in green on this figure. Now, Helicobacter pylori is an interesting exception. It grows in the human stomach, and it can cause stomach ulcers. Hydrochloric acid is present in the stomach, and it creates an extremely acidic environment, and that is part of our defenses. That helps prevent many microorganisms from getting into the body through the GI tract. So the pH of the stomach is around about 2. But Helicobacter pylori is not an acidophile. It actually uses its flagella to burrow down into the mucus layer that covers the surface of the stomach epithelium. And then it also secretes an enzyme. The enzyme is called urease. And that enzyme breaks down urea and makes ammonia, which neutralizes the pH in the immediate environment around the cell. So Helicobacter pylori, it's able to live in that high acid solution and low pH environment, but it is not a true acidophile because it actually prefers a relatively neutral pH in order to grow. It just alters the environment right around it. Like pH, temperature also affects the form and function of biological molecules. Each microorganism has a characteristic range of temperatures in which it grows. This is due to the biochemical reactions that occur within the cells and the stability of the enzymes that control those reactions. The optimal growth temperature, like the optimal pH, is the point at which microorganisms grow the fastest. Okay, so let's look at this green line in this figure. This line represents the growth of organisms that replicate between 15 and 42 degrees Celsius, or in terms that you may be more familiar with, 60 to 107 degrees Fahrenheit. These are relatively moderate temperatures. Organisms that grow best at these temperatures are called mesophiles. Meso from middle. Notice that the growth rate steadily increases from 15 to about 37 degrees, but then rapidly drops off once the cells reach about 42 degrees. There are two reasons for this, and both are related to cellular metabolism. Life depends on a wide array of chemical reactions. As the temperature of a reaction increases, the rate of a reaction also increases. However, it's important to remember that nearly all of the reactions that are found in cells are controlled by enzyme catalysts. Once the temperature gets too high, the enzymes, which are usually composed of protein, denature or lose their shape. At that point, those reactions stop, right? So some organisms are adapted to living at higher temperatures, and they have a higher level of tolerance and stability under those high temperature environments. So this blue line is demonstrating the growth rate of an organism that can grow at higher temperatures. These organisms are called thermophiles or heat-loving. They usually have an optimal growth temperature around about 65 degrees Celsius, which is quite hot. Those that grow at incredibly high temperatures, like those that we see in the hot springs or near thermal vents at the bottom of the ocean, right? So that grow at even higher temperatures than our thermophiles. This is a very clever name. They're called hyperthermophiles. On the other end of the spectrum are microorganisms that can grow in extremely cold environments, even below freezing. So here in gray, you can see an organism that can grow between about -10 to about 12 degrees Celsius. This organism is called a psychrophile or cold-loving organism, right? So it can even grow under freezing conditions, but it cannot tolerate warmer temperatures. There is a special group that is able to grow at very cold temperatures, but also you can see there's some overlap with the mesophile temperatures. This is that light blue line. These organisms are called psychrotrophs, right? They can grow at low temperatures, but also at more moderate temperatures. These are often responsible for food spoilage, even while stored in the when food is stored in the refrigerator, right? They can cause your food to go bad. So speaking of food, the role of temperature in microbial control is really important. Most human pathogens are mesophiles. They're going to grow at 37 degrees Celsius or somewhere around that, right around body temperature. That's 98.6 degrees Fahrenheit if you haven't been following along. And one important way that we can prevent disease, particularly diseases that are transmitted in our food or foodborne diseases, is by controlling the temperature at which we store our food. So cold foods should always be stored below 40 degrees Fahrenheit or 4 degrees Celsius. And hot foods, like you might find at a buffet restaurant, should be held above 140 degrees Fahrenheit or 60 degrees Celsius. Between 40 and 140 degrees Fahrenheit is what we call the danger zone. Those are what are known as permissive temperatures that allow microorganisms to grow. We want to avoid that, right? Following these guidelines, we can prevent the growth of most microorganisms that can cause illness in humans. Of course, I like to talk about exceptions, and one exception is the bacterium Listeria monocytogenes. It can be transmitted in food, so it causes a foodborne illness, and it is a psychrotroph. It is cold tolerant. Sometimes it's referred to as psychrotolerant. It can survive freezing, but it can grow in the refrigerator. And Listeria can be a relatively mild illness, kind of a flu-like illness, but it also can be quite serious depending on if you're someone who might be immunocompromised or elderly. You might get a disease like meningitis or sepsis from this organism. But one group in particular that is susceptible to Listeria infections is pregnant women. It can cause miscarriage and stillbirth in women that are pregnant that get this disease. So it's for this reason that pregnant women are told to avoid foods that are stored for long periods of time in the refrigerator. Those are things like lunch meats, cold cuts, and certain cheeses, right? Because if we store those foods for a long time, even though the growth rate of listeria is slow, it's still growing in that food, and that can cause some serious problems for those individuals. So far, we have discussed the need for water balance and temperature for microbial growth. In the last part of this video, I want to talk about oxygen. The element oxygen is found in all living things. It is part of most of our organic molecules, and it is found in water molecules. Here, we are going to focus on oxygen gas, also known as molecular oxygen or O2. It is used by many microorganisms for growth, but it is not a requirement for all living things. In fact, life on Earth began before molecular oxygen was widely available. The earliest living things were anaerobes, meaning they didn't use oxygen. Cyanobacteria evolved to produce molecular oxygen as a byproduct of photosynthesis. These photoautotrophs completely reshaped the atmosphere of the prehistoric Earth. Eventually, some organisms began using molecular oxygen in aerobic respiration to obtain energy from food, from organic molecules. We will talk about respiration in more detail in the next module, but it is helpful to know that this is a very efficient way to extract energy. Organisms that use oxygen in aerobic respiration are called aerobes. Aerobes grow much faster than anaerobes because of their ability to quickly extract energy from their food. One drawback to living in an atmosphere rich in oxygen gas is the presence of what are known as reactive oxygen species. These are toxic forms of oxygen that can react with and damage biological molecules. Examples of reactive oxygen species include things like superoxide and hydrogen peroxide. Organisms that evolved to use oxygen in aerobic respiration also needed a way to deal with reactive oxygen species. Aerobic organisms produce enzymes that detoxify reactive oxygen species. One example is catalase, which converts toxic hydrogen peroxide to molecular oxygen. We use detection of this enzyme in the laboratory to identify certain bacterial species. So here on the slide you have a picture of a catalase test. It's an easy test that involves adding a hydrogen peroxide solution to bacterial cells and looking for the production of bubbles. So here we've taken a small sample of bacteria, we've smeared it on the slide, and we've just put a drop of hydrogen peroxide, that same hydrogen peroxide you keep in your medicine cabinet, and then it starts to fizz like a soda starts to fizz, right? But instead of carbon dioxide being released, we're seeing oxygen bubbles. Microorganisms can be characterized by their ability to live in the presence of and use oxygen. There are several methods that can be used in the laboratory to classify microorganisms by their oxygen needs. One that we use in our lab here at Valencia is something called fluid thioglycolate medium. This broth medium contains a small amount of agar and a substance called thioglycolate. And these two things together limit the diffusion of oxygen throughout the test tube. Organisms will grow in different locations in the medium based on their oxygen requirements. So I'm going to move from left to right in this picture. Those organisms that must use oxygen, right? They're required to use oxygen. They're obliged to use oxygen. They're called obligate aerobes, right? And they only grow at the top of this medium. Organisms that cannot use oxygen and don't have the enzymes to detoxify it, right? They can't grow around any oxygen at all. They're going to grow at the bottom of the tube. They're obligate anaerobes. In the middle here, we have organisms that can use oxygen in aerobic respiration, but they can also grow without oxygen. Right? So they can go either direction, We call them facultative anaerobes. Notice in that image that the growth is higher at the top of the tube. Remember, I said that aerobic respiration is a really efficient way to get energy out of your food. If you have more energy, you can grow faster. So that's why the growth is denser at the top of that tube. In D on this figure, you have what's called an aerotolerant anaerobe. Notice in this picture that the growth is even throughout the tube. That's telling us that the organism isn't using the oxygen at the top. Instead, the oxygen isn't harming it. So it has the ability to detoxify reactive oxygen species, but it doesn't use the oxygen at all. That's your aerotolerant anaerobe. And then finally, at the very end, in E there, you have an organism that needs oxygen, right? So it's not growing all the way at the bottom of the tube where there's no oxygen, but too much oxygen will kill it. That's called a microaerophile. It can grow in a low oxygen environment. Growing obligate anaerobes in the laboratory requires special equipment, right? Because oxygen is toxic to those organisms. Pictured in the left is an anaerobic jar. So cultures can be placed inside the jar. The jar is sealed, and then you usually will generate a chemical reaction inside the anaerobic jar that will scrub the oxygen from the environment after the jar is sealed. And then you can just stick that whole jar in a regular incubator. Another option is something that I've used, and that's a picture of me back when I was doing my PhD. This is something called an anaerobic chamber, where this whole environment inside that plastic bubble there is anaerobic. There's no oxygen inside that chamber, and I can manipulate the cultures and do tests on the cultures inside the chamber by putting my hands inside these big rubber gloves. So that's another way that we can culture anaerobes without introducing the risk of oxygen. So this concludes our module on microbial growth. In the next module, we will talk about microbial metabolism and some of the biochemical reactions that enable microorganisms to extract that energy from their food and then use that energy in turn to grow.